Before we begin the process of studying glucose transport in the intestinal epithelial cells, it's worth noting this concept of concentration gradients and membrane potential. So here we have a typical cell membrane or a generic cell, and here we have illustrated three ions: potassium ion, calcium ion, and sodium ion. Now typically these ions would migrate to equalize across the membrane. In other words, the potassium ion would be on the intracellular side the same concentration as the extracellular side. Same thing with calcium ion and same thing with the sodium ion. However, you see what's known as a concentration gradient, and the cell membrane actually partitions the inside and outside, and it sets up a situation where ions could spontaneously flow, and the spontaneity of flow begins from high concentration to low concentration. So in a typical cell, potassium ion would want to go from the inside of the cell to the outside of the cell, from where it's more concentrated to less concentrated, to sort of equilibrate across the membrane. Calcium ions would actually want to equilibrate by going from outside of the cell, the extracellular portion, to the inside of the cell, intracellular portion. That is how they equilibrate. And sodium ion would spontaneously want to go from the extracellular side to the intracellular side, that is from outside the cell to the inside of the cell. So we say that all these ions are going down their concentration gradient. They would go from a concentration gradient from high to low. That would be a spontaneous phenomenon. Now, another thing to take into account is the charges. So we not only have positive ions in the cell and outside the cell, but we have negative ions inside the cell and outside the cell. So what's stopping from all the positive potassium ions neutralizing all of the negative ions? Are all the calcium +2 ions coming in and neutralizing all the minus ions? After all, plus likes to go with minus and vice versa. And what's stopping all the sodium ions from neutralizing whatever the negative anions or negatively charged ions are available? Because plus likes to neutralize minus, you don't see that. And the membrane not only sets up a concentration gradient, but it also sets up a charge differential. And that charge differential is called a membrane potential. Typically, as stated here, the inside of the cell is a little bit more negative than the outside of the cell. It actually registers a voltage of about minus 60 millivolts. That's a small voltage, but it's still significant because it tells us that the cell membrane sort of acts like a capacitor. It sort of stores charge. It doesn't dissipate charge. If it were to dissipate charge, we would imagine to be no potential and no voltage registering, because all the plus charges would come in and neutralize all the minus charges and vice versa. So the combination of ions separated by concentration and charges that are separated, pluses and minus separated, both partitioned and both separated by the phospholipid bilayer, that combination is what we call an electrochemical potential. The electrochemical potential is a potential that is caused by the cell membrane, because it's separating charges, positive charges and negative charges, and it's preventing ions that would normally go down its concentration gradient. These ions would want to go down their concentration gradient and also neutralize any type of negative charge that is available. Now the same thing occurs for negative ions as well. A negative ion that's not shown here is the Cl- ion, or the chloride ion. It turns out the chloride ion is a little bit more negative on the inside of the cell versus the outside of the cell. The important point here is we don't get pluses and minuses to neutralize, and we don't get ions to spontaneously go down their concentration gradient. Again, the ability of this phospholipid bilayer to act sort of like a barrier for concentration as well as a barrier for charge. Having given the background behind the electrochemical potential, let's take a look at active transport by the sodium-potassium ATPase. Now that we know about concentration gradients and membrane potentials, we have to also look at this one particular transporter before we go into the relevant and clinically relevant glucose transport by the intestinal epithelial cell. So first of all, let's just parse these words here: sodium, potassium, ATPase. So we're transporting sodium ion and potassium ion, and we're harnessing the power of ATP to do that. So anytime we hydrolyze ATP, we are doing so at the expense of energy. And so what we're doing here is we're doing a transport of sodium ion against its concentration gradient and potassium ion against its concentration gradient. Because normally sodium would like to go down its concentration gradient to the intracellular side, and potassium ion would like to go down its concentration gradient to the extracellular side. But notice what this ATPase is doing. ATP, that energy, is going against what we would normally expect based on the concentration gradient alone. Here we're pumping sodium ions outside of the cell against the gradient, and we're pumping potassium ions inside of the cell, again against the gradient. Also, there's a charge differential. It's not 3 for 3. Instead, we're pumping 3 out and we're getting 2 in. So there is a disparity of charge. We're still maintaining the electrochemical potential. We're still maintaining a charge differential. However, we're kind of keeping things off kilter here, and ATP is the energy provider to allow this to occur. So let's just take a look at this. This is actually a transmembrane protein, and it's going to span the bilayer. So we have the E1 version. So notice that we have one conformation. The conformation binds to 3 sodium ions. We are located inside the cell, and ATP is also bound. So E1 already has previously ATP bound, so the enzyme, or actually the carrier protein, the transporter, has to have a binding site for ATP. And then ATP hydrolysis occurs. The magnesium ion is just there to neutralize the negative charges of the phosphate. So ATP hydrolysis occurs, and you have this sort of transport phosphate intermediate. That phosphate is actually conjugated to the side chain of aspartic acid. That's a high energy molecule with a high energy functionality. So essentially what happens next is that this high energy dissipates its energy by causing sodium ion to go from inside the cell to outside of the cell. So here we're having active transport. We're going against the concentration gradient, going against what we would normally expect to happen based on the electrochemical potential. And now we have an E2 form. The E2 form is just a different conformation of the carrier. The E2 form is still phosphorylated, so that conformation has an affinity for binding potassium ion. It's binding to potassium ion from the extracellular side. And now we have a conformation here. Okay, so let's contrast the E2 form with the E1 form. The E2 form, its conformation is phosphorylated at the aspartic acid side chain and has affinity for potassium. The E1 form has an affinity for ATP as well as 3 sodium ions. That phosphate comes off, and the E2 form now has 2 potassium ions, and it discharges so that the potassium ions come inside the cell, and the E2 form regenerates the original E1 form that is accessible for ATP. So here we have a circuit, and the net result of this circuit is 3 sodium ions from inside of the cell go out, and 2 potassium ions from outside of the cell go in. ATP hydrolysis powers that reaction. It powers that transport, and basically the energy of ATP hydrolysis allows us to go from the E1 conformation to the E2 conformation. We are now ready to actually go into the crux of this topic, and that is glucose transport in the intestinal epithelial cell. So first of all, let's just take a look at a cartoon version of the intestinal epithelial cell. The intestine, or the small intestine, is a site of major absorption of nutrients, and ions and sugars will all be absorbed. If you take a look at this cross-section of the intestinal fold that lines the lumen is just a fancy word for whatever is inside. So what lines the lumen of the small intestine are these columnar epithelial cells known as brush border cells. And these brush border cells on one side of the membrane actually have these projections. So these projections are called villi, and they really enhance surface areas for maximal absorbance. So these villi have actin cytoskeletal features that actually allow them to penetrate up, and once again, it just more area for absorption. So if we actually highlight just one of these cells and blow it up as shown in the figure in your textbook, Voigt and Voigt, you notice that we have one intestinal epithelial cell here. One important point is noticing the differentiation between what is on the apical side, or the side facing the lumen, versus the basal lateral side, or the side that's facing the nervous system, neurons, and the capillaries. This is the side that's really in contact with blood, or the blood capillaries. And this is the side that's in contact with nutrients coming from digestion. So the important point is that we have membrane polarity, or polarizability. The term polarizability just basically means that the intestinal epithelial cell has regions of membrane that are different on one side versus the other side. So you can imagine the different phospholipid components that are on the apical membrane. You can imagine the lipids and phospholipids that line up the villi that allow for surface and nutrient absorption, and contrast that with the type of phospholipids that are here. There is no villi, there really is no surface area maximization. So the phospholipid content will be different from one side of the membrane to the other side of the membrane. So at 180 degrees, we have a different phospholipid composition. Not only do we have polarity, but coming with polarity is not only different phospholipids, but different carriers, and that's another important point. So exclusively on the apical side, you have what's known as the sodium-glucose symporter. So a symporter actually transports two molecules at the same time. Now that symporter has to be localized on one side and one side only. In this case, this symporter is polarized to the apical membrane surface facing the lumen. The transporters here on the basolateral side, or the basement membrane, is going to be very different than what's on the apical membrane. For one, you have a glucose uniporter. The glucose uniporter, as all uniporters, just transport unidirectionally. Plus, you also have the aforementioned sodium-potassium ATPase. Remember what the sodium-potassium ATPase does. It utilizes the power of ATP to transmigrate 3 sodium ions out and 2 potassium ions in. That is against the electrochemical potential. So that is an endergonic process that requires ATP. So with that being said, let's actually focus on what's happening in glucose transport. Obviously, we need glucose, and the intestinal cell needs to transport that glucose to the blood capillaries for our glycolysis if it's a red blood cell, or maybe the intestinal epithelial cell needs glucose for its own metabolic needs or biosynthetic needs. So the sodium glucose symporter, actually situated here, will transport sodium ion and glucose as a symporter. It actually transports 2 sodium ions in, and glucose comes along for the ride. So that sodium ion actually goes down its concentration gradient. So we would expect that to actually be favorable from an energetic perspective. Now, this is a method to actually bring glucose into the cell. Now what happens is that glucose molecule actually gets transported out from the intestinal epithelial cell to the capillary network. And the molecule that does that is this uniporter, glucose uniport. There's a family of glucose transporters. This one belongs to the GLUT2 family. And the GLUT2 family is going to take glucose that's originally came from the diet and transport it to the capillary network. Now that sodium ion that's generated and has come in as an electrolyte goes against its concentration gradient via the sodium-potassium ATPase. So when it goes against its concentration gradient via this ATPase, 3 sodiums come out, 2 potassiums come in. What essentially happens? Well, you're depleting sodium ion, and what will that do to the glucose-sodium symporter on the apical side? Well, the depletion of this sodium ion actually forces more sodium and glucose as its partner in the symporter. It forces glucose to come in. So you're actually setting up a system, or sort of a pump, where by pumping sodium out against its will, against its concentration gradient, you're forcing more sodium to go down its concentration gradient, taking in glucose via this sodium glucose importer. So essentially you're forcing this directionality to go towards more glucose uptake via sodium uptake. And that sodium uptake, by pumping it out, forces more glucose in via the sodium glucose importer, because sodium ions naturally will want to go down its concentration gradient. So this maintains that sodium ion differential, the sodium potassium ATPase. The maintenance of that sodium ion concentration gradient sets up the sodium glucose importer. That glucose goes down and via a specific uniporter goes to the capillary network. The process of glucose going down away from on the basement side to the capillary network, that's known as facilitated diffusion, where glucose would want to go out, but it's facilitated by the process of these transporters, in this case a glucose uniporter. So that is really a nice system that has been developed by the intestinal epithelial cell. It's a trick by Mother Nature to bring glucose in indirectly to the intestinal epithelial cell where it can be utilized, but really it can be taken into the blood capillary network for the red blood cell to use. And what sets this up from an energetic standpoint is the ATPase, using ATP to set up the concentration gradient by pumping more sodium out, causing more sodium on the apical side to come in and force it down its concentration gradient. Notice none of this could happen without polarity. So polarizability is going to be very, very important in this system. Just here, I'm focusing on the bottom left-hand corner. The architecture of these intestinal epithelial cells is very unique. You have your villi, which again are for surface area absorption, and each of these intestinal epithelial cells are really tightly connected to one another. There's a lot of connectivity between adjacent cells. The connectivity is facilitated by many of these transmembrane proteins that span the surface area or the width of the cell, such as tight junctions, adhesive belt, the desmosome, and gap junctions. So many types of enteropathogens, such as E. coli and Vibrio cholera, certain strains of Salmonella, they actually disrupt this. And they break these tight junctions and these connectivities apart, which is one of the causes for the malabsorption that is known when you're infected with these types of bacteria. Another thing worth noting here best illustrates this polarizability. Notice what's happening on the flip side of these membranes. You have a totally different environment, a completely new milieu, where you have the nervous system, neurons, and you have capillaries that are going to transport blood to areas of the tissue or to peripheral parts of the body. This has clinical relevance towards dysentery. So a lot of pathogens that cause malabsorption will prevent absorption of these villi, and mostly one of the causes of that is dysentery. This is a big problem in third world countries where nutrition and access to clean water and just overall hygiene is lacking. So in areas where you do have dysentery, the glucose transport in intestinal epithelial cells gets disrupted. And there is a way to change the system to sort of provide a cure. Now, oral rehydration therapy doesn't really stop the malabsorption. You still will have leakage of nutrients because the underlying villi are disrupted. So it's going to take time to remodel that, especially if you have an enteropathogen or a toxin such as that generated by Vibrio cholera. However, oral rehydration therapy is water with sodium chloride, sodium citrate, a lot of electrolytes, but what is supplemented in these electrolytes? Glucose. So that was a major finding, in fact, that to actually quote-unquote cure dysentery, particularly in the third world, if you supplement your rehydration solution with glucose, you still get some absorption, though a lot of it is still lost. But what you do get is it's just a numbers game. What you do get is glucose absorption. So you lose that weakness because your body can still get glucose and still obtain an energy source. So oral rehydration therapy is not, you would imagine, oh, let's just give electrolytes to the child or to the individual that has dysentery or has struck with an enteropathogen. You have to supplement it with glucose or sucrose, and that forces at least some of the absorption, though you still get some malabsorption if the villi, the underlying tight junctions and desmosomes are in disrepair. So oral rehydration therapy is glucose or sucrose being added to your electrolyte solution, and it forces the sodium glucose symporter, just by sheer bombardment, it forces some of the glucose and some of the sodium ions to get replenished and get absorbed, and it allows this system to actually get back to work. This is an exact same depiction of the previous slide. This is just taken from a molecular cell biology book. Again, notice the polarizability, the basolateral membrane or the basement membrane on one side, the apical membrane facing the lumen or the small intestine on the other side. Glucose is going to come in through the symporter, the glucose sodium ion symporter. The sodium potassium ATPase pushes whatever sodium came in out against its concentration gradient, and that powers the glucose sodium symporter to pump more glucose in. The glucose gets transported via facilitated diffusion via the GLUT family of protein transporters. So again, this idea of this pump is being set up by a concentration gradient maintained by the sodium potassium ATPase and hydrolysis of ATP to keep this pump going. Notice in the blood we have a differential of sodium ion and potassium ion. And notice in the intestinal lumen, we have a differential of carbohydrates, the model carbohydrate glucose, as well as sodium ion. And in the intestinal epithelial cell, notice we have low sodium ion and high potassium ion, but we are pumping against the concentration gradient from low sodium ion to high sodium ion, and we're putting potassium ion in from low to high. These are against the concentration gradients. It is antithetical to what normally these ions would like to do, but the ATP sets it up so that more glucose and more sodium ion can get in. Here's a picture of Robert Crane. This was not really well discovered or well known until the 1960s. And this famous physiologist, Robert Crane, actually discovered the sodium glucose symporter, and the discovery of that was actually key and landmark in oral rehydration therapy, putting glucose into these electrolytic solutions, whereas people prior to that were just giving salt solutions and salts and replenish, but they were lacking the glucose. Once the sodium glucose symporter was discovered on the apical side of the intestinal lumen cell via Robert Crane and others, really the diagnostics and the treatment of dysentery really changed. So here's a diagram of his sodium glucose symporter that he drew on a napkin just before he were to give a talk at a physiology conference around 1960 or so.